Thanks for viewing my talk today. The anthropogenic emissions of greenhouse gases have had widespread effects on global climate, including increasing temperatures, alteration of precipitation regimes, and increased frequency of extreme climatic events. As these changes are expected to continue and worsen throughout the 21st century, these climate shifts pose a serious threat to global biodiversity and ecosystem function. It is clear that these changes will have widespread ecological impacts, as studies have already documented impacts both across taxonomic groups as well as biomes. Species can potentially respond to these climatic changes in one of three main ways, either through phenotypic plasticity, evolutionary adaptation, or through shifting their ranges to track their moving climatic niche. If species are unable to effectively track these changes through one of these methods, they are likely to face extinction. Today, I will specifically be focusing on species' response to climate change through range shifts. As rising temperatures are one of the most widespread effects of anthropogenic climate change, it was initially predicted that species would shift to higher latitudes and elevations as temperatures rose to track their climatic niche, as illustrated in the figure on the left. However, as more responses have been documented, it is clear that species responses are much more idiosyncratic than expected, with some ranges shifting while others contracting or expanding, as shown on the right. Other abiotic factors, biotic interactions, as well as barriers to dispersal, colonization, and survival can all play a role in determining the direction and magnitude of the shift, resulting in these varied responses. Understanding how trees will respond is particularly important in the western United States, as this area is expected to experience dramatic shifts in precipitation and temperature. When evaluating range shifts in long-lived sessile organisms such as trees, you can use life stage as a proxy for time to evaluate how future distributions will differ from current ones. In all of these next figures, the red symbolizes the adult distribution, while the blue curve symbolizes the seedling distribution. If a species will occupy the same range in the future, you would expect regeneration to be occurring in the same area as the adult distribution, as shown in that first figure. However, a shift can be indicated by adults in the absence of seedlings at one edge and seedlings in the absence of adults at the other edge. In the case of a northward shift occurring as a result of species tracking the warming temperatures, you would expect seedlings at more northern elevations or latitudes than adults, while regeneration being absent from that southern range limit. Inversely, a southern or downward shift is predicted when seedling regeneration occurs at the southern or lower range limit beyond the adult distribution. And this type of response has been found to occur due to tracking changes in water availability at lower elevations. Range contraction is predicted when regeneration is only occurring in a subset of the current adult range, suggesting that the distribution will shrink in future generations. As communities reshuffle and other species shift, new biotic interactions may restrict a species from regenerating throughout the entirety of its historic range. And finally, expansion is indicated by a seedling distribution beyond the current adult distribution at all range margins. And this can be caused by an interplay of environmental factors and potentially novel biotic positive interactions, allowing for colonization of new areas. With that idea in mind, we developed the following two questions. Do adult and seedling distributions differ in western U.S. tree species, and are the differences in seedling and adult distributions indicative of range shift, contraction, or expansion? To evaluate these questions, we use the forest inventory analysis data for California, Oregon, and Washington, collected by the U.S. Forest Service. This data set represents a systematic sampling of all the forested area within the U.S. with about one plot per 2,400 hectares. Throughout California, Oregon, and Washington, there are over 50,000 plot locations. Of these 50,000, only some are forested, and in those forested areas is where the plots are actually inventoried. And so each plot that is inventoried consists of 4 circular subplots, each with a radius of 24 feet, where all adult trees are inventoried. And within each subplot, there is a smaller 6.8-foot radius microplot where seedlings are recorded. In addition to the FIA data, I used 15 different raster datasets of environmental predictors to inform my species distribution models. These variables included elevation, temperature, precipitation, vapor pressure deficit, soil water balance variables, and soil data. To go through the methodological process I will use the example species of Quercus was Lezennii. So for each species, I converted that FIA data into presence-absence data for both the adult and seedling life stage. I then extracted the environmental variable values for all 15 variables shown on the previous slide for all the presence and absence locations. I then generated two generalized linear model species distribution models, one for the adult and one for the seedling life stage as shown to produce these probability distributions on the figure on the right. From there, I then compared the adult and seedling distribution models by calculating the niche overlap and the difference between the two species distribution models. We then extracted the latitude and elevation data from the raster, converted it to point data, and compared the latitudes and elevations of adult and seedling only presence for each species. Here are three examples of the types of responses I found across the 25 species I evaluated. For AB's concolor, there was no significant difference between the adult and seedling distributions. In these maps, yellow indicates both adults and seedlings being present, while red is adults in the absence of seedlings, and blue is seedlings in the absence of adults. Next, Alnus rhombifolia showed signs of expansion and shift as indicated by the blue northern edge. You can also see that some contraction is occurring at that southern range edge as well. Finally, for Calycedrus decurrens, there is strong signs of contraction with seedlings occupying only a fraction of the adult range as indicated by those red cells. Overall, of the 25 species examined, 18 species had seedlings that occupied higher latitudes and 10 species had seedlings that occupied higher elevations than adults, both suggesting future shifts towards cooler climates. However, we found that for 12 species, adult distributions existed at higher elevations than their conspecific seedlings, and for 10 species, adults existed at higher latitudes than their seedling counterparts. Finally, for 3 species, each showed no difference in latitudinal or elevational distributions between life stages. This high variation response across species drives the question, can we predict species responses? Functional traits, which are characteristics that are expressed in phenotypes of organisms that determine how these organisms will respond to their environment, may be a way to explain and predict species response and ultimately create a trait-based predictive framework to inform conservation efforts. Specifically, dispersal traits likely play an important role in a species' ability to shift its range as it determines the species' potential to match the shift in their climatic niche, and species that can disperse for longer distances and at faster rates are more likely to be able to make that shift in the appropriate timeline. We specifically evaluated the ability of seed dry mass and dispersal mode to predict species responses. To do this, we used trait data collected from various online databases to determine the primary dispersal mode for each species as well as the average seed dry mass within the area within the United States. This figure shows the relationship between seed dry mass and latitude difference between adults and seedlings, with higher values on the y-axis indicating a northward expansion and anything below 0 indicating a northern contraction. As you can see here, there's no significant relationship that exists between seed dry mass and species range shift response. Similar to the last figure, this figure shows the relationship between elevation difference and seed mass. Seedlings occupying higher elevation than adults was positively correlated with increasing seed dry mass. This figure shows the latitude difference compared to the 3 categories of dispersal mode: animal, wind, and gravity. Both animal and wind dispersed species showed greater signs of northern expansion of seedlings than gravity dispersed seeds. This figure shows the elevation difference relative to dispersal mode. While once again animal dispersed seeds showed the greatest degree of magnitude and elevation difference, none of the dispersal modes significantly differed from 0. In conclusion, adults and seedlings of western U.S. tree species occupy different latitudinal and elevational ranges, suggesting future changes in distribution. And these responses varied quite a bit across the species. And seed dry mass is not always a good predictor, as higher seed mass was correlated with seedling expansion to higher elevations, but not latitudes. And finally, animal dispersed species may be more likely to track their shifting climatic niche to higher elevations and latitudes than wind or gravity dispersed species. Thank you so much to everyone who helped with this project, and thank you for taking the time to listen.